The reason why I'm talking about alcohol use is not only because you guys enjoy the alcohol here at the meeting. It is because actually it has been a long interest of my research group. So I don't know how much you know about it, that the alcohol use actually is growing tremendously throughout the entire globe, actually. About 400 million people, about 7% of our world population of 15 years or older suffer from alcohol use disorder. This is very recent WHO data. And the alcohol-related death is increasing, and one of the 10 leading causes of death is disability worldwide, and it's also is a major risk factor for cancer that actually I'm not going to talk about today. And needless to say, this costs a lot of money to the entire US economy and all of us. So I think the most important thing to me is still in alcohol use and alcoholism is that, you know, as in recent years, we so much talk about mental health and accepting that, you know, people are sad or maybe depressed and then sensitized to ask about it. But when it comes to alcohol, I think it still carries more of a stigma than IV drug use or any kind of other addictive diseases. And I think this is something that many of us can kind of consciously improve, even in fields that are not necessarily a psychiatry kind of setting. I'm a hepatologist, a gastroenterologist, and I think this is an area that I think our field is kind of recognizing that it's not enough to fix the liver if the alcohol use and the habits are not necessarily improved. So if you look around the world, this is data from 2019. The green kind of shows, the darker green indicates more alcohol consumption around the globe. So I'm sure that everyone is looking at, where is Hungary? It's somewhere in there, kind of is amongst the actually leading countries, I think. And and you know, chronic alcohol use leads to multiple different kind of diseases. But as a hepatologist, I'm interested in liver scarring, that's called cirrhosis, and in fact that again there's more than 10 deaths per 100,000 people, and you can see that Russia is kind of leading in that, but I think Hungary is like somewhere here, so it's kind of up there. We are up there. And then in the US, alcohol-induced liver disease actually became one of the leading causes of liver transplantation in the past 5 years or more. I think that just shows that, in fact, in the United States, that's also a kind of a major issue. So then one can ask that, you know, what is a drink, right? You have, some of you had this much, some of you had that much, some of you may have, I don't know, multiple ones. So according to the American, to the National Institute on Alcoholism and Alcohol Abuse, that kind of standardizes this thing. In the US, we talk about 14 grams equivalent of alcohol per drink. It's interesting because in Europe, typically it's only 10 grams, but, you know, here it's different. And that would be like one beer, and then about 5 ounces of wine. And don't ask me how many milliliter is that. I always have to look it up. And then the same thing with the spirits. So why is this important? Because when we kind of want to do any kind of studies, obviously we want to quantify the alcohol use, which by the way is very difficult because people may not remember or may not want to remember or may not necessarily want to, you know, really face the fact, the amount of alcohol use. So some of these studies all has the caveats, I think, when it comes to that. But we defined it in a way that what is moderate drinking, and according to the US guidelines, it's about 2 drinks or less for men and 1 drink or less for women. And the reason for that is because there is a gender difference in that. So as much as I kind of think that there should be a lot of gender equity in pretty much every part of our life, unfortunately, when it comes to alcohol, there's no gender equity because there are multiple studies that show that females actually are develop liver disease and have a more sort of aggressive liver disease development with alcohol use if you look at the same amount of alcohol as compared to men. And I'm not going to go into the details of it, partially because I think the explanation for that is not really very well understood. There are certain hormonal factors and all kinds of other things have been suggested, but I don't think that we really know why. And this kind of goes along if you look at the binge drinking kind of cutoff. So for men, typically it's identified as about 5 or more drinks in a matter of 2 hours. So binge drinking means that, drinking heavy for a shorter period of time and really getting drunk. And it's important because I think the alcohol levels, depending on how high, they have potentially different biological effects. We don't exactly know all the differences, but I'm going to show you some data that it probably has a major role in that. And then there is heavy drinking. That's more like a continuous chronic alcohol use, 5 or more drinks on any day, or 15 or more drinks per week. And again, here are the differences between the genders by the definition. So what happens when we drink alcohol? Well, obviously, kind of most humans drink the alcohol and that doesn't come from other sources. So obviously there is a higher concentration in the upper GI tract, in the esophagus, stomach, and recently actually we realized that it's also very high in the proximal small intestine, and in fact has a lot of effect there. And then through the intestine, it goes into the liver through the portal circulation. That's not really shown here. And it is, for a long time, it was postulated that the alcohol metabolism happens in the liver, but actually it happens in many other cell types and then potentially in other organs. Certainly there is some even in the stomach, but the majority happens in the liver, and then it happens in a way, and I'm not going to go into the chemistry and the biochemistry of this, don't get scared. The alcohol, ethanol up there gets metabolized actually pretty fast by an enzyme called alcohol dehydrogenase, very fast to acetaldehyde, which is kind of a toxic compound, but that's also very short-lived, and it's metabolized further by aldehyde dehydrogenase into acetate. And these enzymes that participate in the breakdown of the alcohol actually have some subtypes, isoforms, and that could be genetically determined. So, for example, Asian people tend to have the versions of the alcohol dehydrogenase or the aldehyde dehydrogenase that causes that doesn't work very well, and then it accumulates these kind of compounds for a longer period of time, and that these are the ones who kind of get a little reaction in like being very red or unwanted kind of side effects. But overall, that's the way that the breakdown happens. So what happens also that after a certain threshold of alcohol use, then other enzyme systems kind of kick in. And that would be catalase or this microsomal alcohol metabolizing enzymes. And then once those are involved, then all kinds of reactive oxidation species, so kind of, again, bad mediators happen that can essentially negatively affect and be toxic to cells. So I think that kind of goes to the kind of message that the smaller amount of alcohol, we are very well equipped to handle with this enzyme system, but when the system is overloaded, then there is like all kinds of byproducts that are toxic. And in fact, we know that alcohol affects multiple organ systems. So each one of us, the neurologists, are interested in the brain effects of alcohol, the gastroenterologist and in the liver, and then of course there are some other people, other organs. But the bottom line is that the alcohol in the circulation, in the blood alcohol levels go up, and it affects every organ. And in fact, there's one common element in that we were very interested in this, which is inflammation. That you may have heard about that in other diseases, because that's kind of is happening in many other conditions too. But certainly this is something that we have discovered and been very interested in the context of alcohol use, particularly in chronic alcohol use or large amounts of alcohol. So I'm going to show you a little bit about what happens in the brain, liver, and intestine with alcohol use, and also what kind of mechanisms are involved, just to give you a kind of a flavor of the kind of things we do. So many years ago, actually, we did this study that we took healthy volunteers and invited them to get drunk. So my husband was the first subject, but maybe I shouldn't say this, right? He did this 2 milliliter vodka per kilogram body weight alcohol consumption, which makes people pretty drunk. So the legal limit in Massachusetts is 0.08 gram per deciliter. And so with this, people kind of get drunk, get to 0.1, even some people was a little higher than 0.1. So we did all of this in a clinical research center under controlled conditions, and give, people didn't drive. Even the next day, we sent them with like a taxi and stuff like that. But what we did, we also took blood from these individuals every half an hour for a period of 4 hours when they were in the clinical research center, and before the alcohol consumption. Now, the kick was that they have to drink this thing in half an hour. So there's clearly a binge drinking. I think in those days, they didn't necessarily call it that much of a binge drinking. But anyway, this is what we found. Serum endotoxin. So this endotoxin is a component of gram-negative bacteria from the essentially intestine, from the stool and whatever is in the intestine. And those levels went up very high and came down. And by 24 hours later, they were actually normalized. This is done here. The serum 16S DNA is a marker of bacterial DNA. So it means that certain components of the bacteria actually got translocated too. And this was after one occasion of binge drinking. So now if you look at patients who get admitted to the hospital with alcohol-associated hepatitis, so this is like they are very sick, but they are sick from the alcohol-induced liver damage. Then we look at the endotoxin that's like so high compared to normal controls. And these guys don't drink alcohol anymore because they are in this hospital and they're too sick to drink. The same thing, the plasma 16S is high. So it indicates that there is something going on. And in fact, what we know from multiple studies, that alcohol, as I told you, affects various tissues. So once somebody drinks alcohol, there's a whole bunch of things happening in the gut, so in the intestines. And this is a snapshot of the intestine. So this is the lumen, where there is these things are the bacteria and viruses in the poop. And this is the epithelial cell layer. And in normal individuals, this epithelial cell layer is very tight. So we don't get fever from having food or having the microbiome in our gut. But what alcohol does, multiple things. One is that it actually impairs these tight junctions, so it becomes kind of leaky. And that induces all kinds of immune cells to come to the small bowel or even throughout the bowel. But then it also changes the composition of the microbiome. And so I'm not going to talk about that, but recent studies really show that certain types of good and bad bacteria balance actually changes with chronic alcohol. In fact, viruses that live in the gut and fungi kind of change. So there's a lot of effects of alcohol that happens. So why is it important? Because all the blood that goes into the intestine gets collected through the portal system. So this is the veins that bring back the used alcohol, not alcohol, the nutritional components, for example, after we eat. And it comes back to the liver. So the liver is kind of almost like a filter. And takes up all these good nutrients and helps us to do energy and all those kind of things. But in the setting of alcohol, all these bacterial components come to the liver and can induce inflammation. At the same time, alcohol directly damages hepatocytes through some of those breakdown products of the alcohol metabolism. And all of this kind of goes on in an alcoholic individual for a long time, and in fact induces scarring that's called fibrosis in the liver. And interestingly, very similar mechanisms happen in the brain. The brain immune cells, the resident macrophages called microglia and astrocytes and recruited macrophages are activated and inflammation happens in the brain. That recently has been suggested that actually fueling the addictive behavior in alcohol use. So, oops, I'm going backwards. So the common elements in the pathophysiology, we believe both in alcohol liver effects and in the brain include some of this direct cellular damage that alcohol causes, also change in the gut permeability induced microbiome components and innate immune cell activation. So what we do is then go to the mice and see if we can mimic certain conditions. And so here, mice were fed for whatever 42 days with alcohol or without alcohol. And actually, mice like alcohol a lot. And girls and female mice like it more than men. But that's a different story. So anyway, I'm not going to go through the details. The bottom line is that if you look at infiltrating macrophages, so macrophages like the first kind of type of immune cells that can migrate to the site of any kind of infection, and they play a major role in host defense. And these go into if you give them alcohol, then in the brain it actually goes and they become enriched in there. And the microglial doesn't change too much, but the infiltrating macrophages do. So then we ask the question that what happens if we block this macrophage recruitment to the brain or anywhere, and we use this compound called salicruvirol, which essentially is an inhibitor of the receptor that can kind of call for these macrophages, the chemokines. And what we found was that if you give this TVC, then the alcohol effect is actually attenuated, and it is attenuated whether we start to give it from the very beginning of alcohol use or if we just give it later, kind of mimicking that now we have an alcohol-induced inflammation and can we kind of stop it at that point? And the answer is yes. And in fact, that also reduced the inflammation. This TNF, the IL-1 and IL-6 are inflammatory cytokines that are kind of readouts for all this inflammation. So that was in the brain. And in the same mice, we also looked at the liver. So what happens is in the liver, alcohol induces fat deposition and also liver damage. So when we look at ALT or that's one of the transaminases in the liver. That's something that we look at like in clinic to check if somebody's liver is kind of normal or maybe affected. And in these mice, the alcohol with the open bar, open circles, causes a major increase in these transaminase levels. But in a minute, we start to give the salicruvirol here. It goes down to normal. This is very impressive. And again, this is in the mice. Macrophage infiltration and inflammation was also reduced, and we found reduction in the fat deposition. So we were this close to convince the company to do a clinical trial with this compound when the company got bought up by another company. And of course, they decided that now they're not interested in this anymore. So end of story there. But what we learned from these experiments is that chronic alcohol consumption actually promotes the recruitment of these macrophages to both the liver and to the central nervous system and promote inflammation. And if we inhibit the recruitment of these macrophages, actually we can attenuate the effects of alcohol. So I showed you earlier how things coming from the gut can fuel the inflammation. So Patrick, who was an MD PhD student in my lab, asked the question, what happens if we kind of decontaminate the gut? And we didn't do like an absolute decontamination, but we gave this heavy-duty antibiotic combination, which essentially brought down so by 5 days in the antibiotic, we didn't get any bacterial colonies growing. And in fact, the antibiotic continued to attenuate the kind of colonies in the gut, even in the presence of alcohol. And what we found was that if we do the antibiotic treatment in the gut, then the alcohol-induced serum endotoxin levels were reduced, and the bacterial DNA that I showed you before induced by alcohol also attenuated. So essentially, it looks like that these bacteria actually do play a major role. And if you further look at the inflammation markers in the various tissues, liver, small intestine, or in the brain, the same thing is that in the presence of alcohol, antibiotics, we could attenuate the inflammation caused by alcohol. But the interesting thing was that this gut sterilization improved the liver steatosis and also the inflammation markers here, but it didn't really have any effect on the liver damage or the hepatocyte kind of damage, this measured by the transaminases, suggesting that the alcohol metabolism itself can still damage the hepatocytes, but maybe the inflammation is reduced. So overall, that's a positive effect, but not enough to completely stop the effects of alcohol. So this suggests that alcohol-induced pathogen-associated molecules increase the inflammation, and at the same time, the hepatocyte damage continues in the presence of alcohol. So why study the liver? Well, I think the liver is kind of a special organ, and liver disease actually can be a silent killer. So in the Greek mythology, Prometheus was one of the characters, he probably Zeus's son. And he created, according to Greek mythology, humankind, and then his brother created the animals. And turns out that the animals had fur, and humans were naked, or whatever, not naked, but yes, naked and no fur. So humans were very cold at night. And then so Prometheus took pity on them and essentially gave them fire. The problem was that fire was the special tool of the gods. And when Zeus found out that this happened, then he got incredibly angry at Prometheus and wanted to punish him. So his punishment was that Prometheus was chained to the mountain, and essentially this eagle, which doesn't really show very well, it's kind of dark, this Rubens painting. The eagle came every night and picked on Prometheus' liver, and the eagle actually was, Zeus was in the form of eagle, but then Prometheus continued to live because the liver regenerated every day. And in fact, we know that the regenerative capacity of the liver is really powerful. Now we take advantage of that in the liver transplantation, that we can take out 2/3 of a liver of a normal person and then give it to a recipient, and both of the livers regenerate to 90% of the original size in about 6 weeks. So it's really, really, really amazing. So what happens is that the normal liver, however, can go into this sort of cirrhotic and damaged liver, and that doesn't regenerate anymore. In fact, during this kind of change, a lot of regeneration capacity changes. And we know that inflammation and chronic liver disease leads to that. Various types of viral infections, and as I'm showing you today, alcohol. So because of this, the natural history of alcohol-associated liver disease is in a way that it can go very quietly without really giving much or any particular signs to the individual, and lead to fibrosis and cirrhosis. And very often we find patients we come across who are already in this later stage. But this is why I think early detection and in primary care setting, looking for this is so important, because it's reversible. The steatosis stage is absolutely reversible, and probably the steatohepatitis is also something that can go back to normal because of the regenerative capacity of the liver. What happens is that if there's a lot of alcohol use, and not in every individual, but you often see this alcohol-associated steatohepatitis picture, when people come in being jaundiced and very, very sick, and they get admitted, and in that stage, there's they're very, very sick. And certainly this causes all kinds of, cost and readmissions, and it's a major problem for the individual and also for the healthcare system. Okay, so I think we have okay, I think sorry. Okay, so here, this jumped. Okay, so what happens is that this alcohol use and the mortality due to alcoholic cirrhosis actually is rising, and in particular in the US, it is rising. And look at this age group is 25 to 34, where we see the biggest jump in the cases. So I think that's very alarming, and we know that young women actually drink more, and young women actually probably have more of this alcoholic hepatitis than guys. So it's a message that we need to get across. So if we look at alcohol-associated hepatitis, we talk about moderate and severe, and this is like a scoring that we use, but this is also a scoring that is used in liver transplantation, and typically 20 or higher is the one that people actually can qualify for liver transplantation. In reality, they really don't get transplanted, especially not in the New England region, until they get closer to 40. But the important thing is that if you look at the 90-day mortality, it goes up like a really exponential curve. And these people who have this severe alcoholic hepatitis, essentially their survival rate is lower than many people with like stage 4 cancers, many cancers. Yet our treatments are very limited. I don't know why is this not doing stuff, but anyway. So if we look at the therapy, this therapy that we have with corticosteroids was established in 1970s, and there is no new drugs for this disease since then. So I think don't have to convince you that we need new therapies, because it really is not giving a major survival benefit over more than 28-day survival, but the long-term survival is not improved. So for this reason, we thought that we need to look into what are the reasons and the components of the pathomechanisms, because maybe targeting that could help us to come up with better kind of interventions. And knowing the components that all of these play a role, we were interested in prediction factors. So I think I'm going to skip this. But with the inflammation, we know that the inflammation is happening through recognition of these various damaged molecules and damage-associated molecules from cells, and also the gut-derived pathogen-associated molecules through what are called pattern recognition receptors that can distinguish between endogenous and exogenous danger signals, and also respond in a very kind of similar way by inducing inflammatory cytokines. And in fact, this is a pathway that we were very interested in, and many others in case of alcoholic liver disease. And I just couldn't help myself to show that we were very excited about it, because these were the pathways we worked with and collaborated with, particularly Bruce Beutler, who gave us some of mice and everything. And essentially, they got the Nobel for this in 2011 because it's really so profound. The other one is Jules Hoffman and Ralph Simon, who worked on dendritic cells. And I remember that when I was the first postdoc in the Netherlands, Ross Simon was the invited speaker in our little lab, and then he was talking about these dendritic cells that that was the discovery of the whole thing, actually. This was back in like late 80s. But anyway, what happens is that in the context of alcoholic liver disease, we know that these receptors, these evolutionary conserved receptors, play a major role. And the interesting thing is that these toll-like receptors actually are 98% preserved between the Drosophila and humans. So this is really the first line of defense and recognizing danger signals. And there are intracellular and extracellular sort of components of this. And we particularly were interested in and then discovered this intracellular component that's called inflammasome, which is a multi-protein complex, and has various components that this actually can get out of the cells. And the reason why we were interested in this, because in alcohol-associated hepatitis, people stop drinking. They get admitted to the hospital, but then they get sick and they have this inflammation going on for weeks and weeks and weeks. And there's really no good explanation that why is that happening, this sustained inflammation, when they actually stop drinking. This is obviously not just the alcohol alone. So what we found is that there's components of this inflammasome that can induce inflammation and propagate inflammation actually get out of the cells. And in fact, if we give these, we called SPECs, into naive mice, that induces inflammation. And in fact, it induces inflammation in the liver. So essentially, we showed that we can block this through small molecule inhibitors. And again, this is in the mice. It's not in human clinical trials now. But I think it's kind of interesting and just shows that if we intervene with some of the components of inflammation, actually, we might be able to attenuate the disease. And one of the kind of effector molecules that this inflammasome has is the caspase cleavage that allows interleukin-1, which is a major inflammatory cytokine, to come out from the cells. And then so we were interested in the role of this interleukin-1 in liver disease, in the alcohol-induced liver disease, because we know that it has multiple sort of effects that relate to the liver disease with inflammation, hepatocyte death, regeneration, and liver fibrosis. So we asked the question that what if we stop this or block this pathway? And there is this recombinant, there is an IL-1 receptor antagonist that's naturally occurring, but this is also produced as a drug and being used in various rheumatoid diseases to reduce inflammation. So we asked the question whether blocking IL-1 could help. And then that kind of led us to a clinical trial, in which it is a multi-center clinical trial where we combined the anakinra, which is the IL-1 receptor antagonist. We used pentoxifylline that can stabilize hepatocytes, and zinc that somewhat improves the gut permeability, and use this in a multi-center double kind of blind placebo in the pilot study where we compared prednisolone, the standard of care, with this combination of drugs, and in patients with alcohol-associated hepatitis. And essentially what we found was that, well, if you look at the first 28 days, the survival was very similar between the two groups. So we did not necessarily have a miracle drug or miracle combination, but at least at the long-term survival, there was a kind of early signal that potentially this combination is good. So additional clinical trials actually were initiated, and this was a multi-center clinical trial supported by the NIH. So another component in alcohol-associated liver disease is the fact that neutrophils that are, again, another inflammatory cell type are really abundant in the liver. And that's very unusual compared to other type of liver diseases. So we were asking the question that, why is that and what is the role of these cells? Typically, these cells actually play a role in inflammation. So if you have like a zit on your face or any kind of inflammation, this is the cell type that goes there first and really cleans up the sort of infection. But these patients actually are susceptible to infection. So it's kind of like a controversy that we have all these cells in the liver, yet the patients are dying of infection. So we started to look into the various subtypes of these cells and discovered that actually the neutrophils are not like all homogeneous. We have various populations. And then when we did like gene sequences of those, then we find that what we call high density neutrophils are very much activated and produce a lot of inflammatory responses, whereas the other ones that are low density neutrophils are exhausted. But they're still around and they don't die. And in fact, they stay in the liver. So then my graduate student actually was going further into this question, and essentially she found that, so if you look at the blue stains, those are DAPI, so the DNA in the cells is stained. And if you look here, if you give alcohol an hour later, the DNA is coming out from the cells. So these neutrophils can do this, like neutrophil extracellular traps that are really enriched in DNA and saturated histone. And in fact, in bacterial infection, these are designed to trap the bacteria in the tissue without letting them to go all over the place. So what happens is alcohol kind of does the same thing, and that's not necessarily good because it damages the liver at the same time. So anyway, she kind of discovered really nice that this mechanism contributes to these low density neutrophils that now are surviving, and they're studying this further to figure out how much they actually contribute to the increased susceptibility to infections. And then I'm not going to go into details, but just to give you an idea that in mice, we showed that actually if we deplete the neutrophils, that you can do in mice, but don't want to do in humans, that kind of attenuates disease, or we can actually disrupt these nets or inhibit NLRP3 that actually stops this entire process. So I'm just going to spend a few minutes at the end to talk about this other sort of line of investigation we do in alcoholic liver disease, which is related to micro-RNAs. So these are intracellular RNA types that are 22 nucleotide sequences, and they're non-coding, but they have a lot of effect because they are produced in the nucleus and they come out to the cytosol, where they can have multiple effects. They can target various messenger RNAs by binding to the appropriate kind of matching sequence of RNAs, and then modulate the expression of the target RNAs, and it can also do translational regression or deacetylation of messenger RNA. So we were interested whether these have any role in liver disease, and in fact found that various cell types express various types of microRNAs, and very uniquely microRNA 122 out of the 900 plus different kind of microRNAs. The 122 is the one that is uniquely really abundant in hepatocytes. So it turns out that about 80% of the microRNAs are microRNA 122 in hepatocytes. And that kind of an abundance is not known in any other cell type. So it's not really understood why, but it's very curious. But what we found is that this miR-122 actually in the hepatocytes goes down with alcoholic liver disease. And that probably contributes to some of the differentiation we see in these cells. We also identified microRNA 155, that's a regulator of inflammation. So in the microRNA 122, we find that it actually in the hepatocytes can induce inflammatory pathways, but interestingly, it also comes out to the serum. And it is one of the very early markers of liver hepatocyte damage in particular in any kind of hepatocyte damage. So acetaminophen-induced injury or alcohol or non-alcoholic fatty liver disease, we find that. So in addition to that, the other microRNA is very interesting is microRNA 155 that's more abundant in immune cells. And we also found recently that it actually has a role in liver fibrosis, so in the scarring process. And now we are doing various experiments to design how could we block this or inhibit the miR-155 as potentially a therapeutic target. But I also wanted to show you that earlier on, this was back like 10 years ago, when we worked on this microRNA kind of project, then I was at UMass, the University of Massachusetts Medical School, and we were talking with Victor Ambrose, who is the one of the two people who discovered microRNAs. And Victor is a chemist and he's not a biologist, so he was like absolutely fascinating and drilled me on that. So okay, so if there is this microRNA, does it go to the liver? Does it go to the brain? Where does it go? So it's like, okay, well, let's do the experiment. So we did this experiment where we had microRNA 155 knockout mice. So these mice didn't have miR-155, but then we knew that we can, in wild type mice, there is a lot of miR-155 and we can enrich it. And in fact, enrich it in exosomes. So we shot this, we gave this microRNA 155 mimic to this miR-155 knockouts and looked for the distribution. And then very fast, this was injected intravenously in mice, so it obviously went to the liver. So in 10 minutes, it was in the liver, but it went to the adipose tissue and to the lung and to a certain extent to other cells as well. And the liver actually ended up in hepatocytes and also in mononuclear cells. So this was kind of a really interesting way to show that actually the biodistribution of these microRNAs happens fast. And it happens even if you put them into these little exosomes. And I was so excited to see that they got the Nobel this year. I know that UMass was waiting for this for a long time. So anyway, and because of this whole microRNA story, we became interested in the extracellular vesicles that could be various sizes. So these are small membrane-coated kind of products that come out from cells, little kind of bombs, if you will. Some of them are through a kind of active secretion mechanisms. Those are exosomes that in the kind of 50 to 150, 120 nanometer range. But there are some larger microvesicles that can come out of the cells. And essentially, we are looking at this as a potential biomarker, looking at their cargo, because it turns out that the various stimulation of cells can change the cargo in these vesicles. We also find that these vesicles actually do intercellular and even inter-organ communication. So this is kind of another area that we kind of doing. And this is an earlier study that we showed that if you look at the hepatocyte here, and put alcohol on them, they start to produce these little extracellular vesicles. And then we showed that there's a lot of interaction between the hepatocyte-derived extracellular vesicles and immune cells and so forth. So it's kind of fascinating. So to end, I wanted to show that, alcohol-induced liver disease is just one kind of steatotic liver disease. And I think you probably heard others speak here. Dr. Buffy probably gave talks about the steatotic liver disease, which is it's called NASH in the past, but now there's a new name. This is metabolic dysfunction associated steatotic liver disease that goes along with risk factors of, obesity, metabolic disorders, insulin resistance, and hypertension, and really is one of the major causes or additional causes of liver disease nowadays. And it's not only alcohol. And in fact, if you look at the US map between 94 and 2015, with the darker red being the frequency of the obesity greater than BMI greater than 30, you get the sense how there's a tremendous increase. And that, of course, goes along with the potential to progression of liver disease. And this is not only the US, because we know that the global prevalence of obesity is also increasing. And the type 2 diabetes kind of goes hand in hand with that because obesity increases the risk of type 2 diabetes. And this leads to a high burden of this metabolic dysfunction associated with steatohepatitis. And currently it is on the right upper quadrant. You can see that the Western Europe has about 20% prevalence. East Asia has 29%. And so this is certainly not just the US. And it also leads to the increase in burden of death. So based on the studies that we did, and I'm not showing you all the details related to the metabolic dysfunction associated with liver disease, but a lot of components that I showed you in alcohol is happening, maybe in a little lesser extent, but the same pathways actually are activated in this metabolic associated liver disease. So now it becomes the question that what happens if both of these are happening in an individual. And in recognition of that, actually, the societies, the American Association of Studies of Liver Disease, that I personally had the privilege of being the president of 10 years ago, and also the European Liver Society came up with this combined sort of guidance now, and we recognize that this metabolic associated liver disease can be overlapping with alcohol use. And in some people, just a little alcohol use or more alcohol use. And this is like a hot area now to figure out that how to design clinical trials in this, how to define all of this, and how to help our patients to really kind of minimize this disease. But in fact, obesity sensitizes the liver to alcohol-induced damage because here, if you look at this thing, so it shows the alcohol drinks 0 to more than 2, and the BMI, that's the body mass index, going from normal to increase. Then look at that, the red one. So BMI 25 to 30, which is like really not that obese at all. But if they drink more than 1 or 2 drinks, then the increased liver transaminases are much higher. Or if you look the other way around, if 1 to 2 drinks per day, but now somebody has a BMI greater than 30, then very likely that they're going to have abnormal LFT. So overweight and obesity increases the risk of alcohol-related abnormal transferases. And actually, it increases the liver-related death too. I just didn't include that. So needless to say, this is a hot area of liver treatment discovery, and all the drug companies that were in the liver field now are looking at the MASH potential drugs, and they're not going to go into details of this, but all of these are like more than 200 clinical trials going on. I think it's very encouraging because now if I look back on the greatest story of translational science and medicine in my lifetime, which is hepatitis C, I think we really have a lot of hope because hepatitis C is a virus that affects the liver and caused all this cirrhosis and essentially a lot of death. But until 1989, it was called non-A, non-B hepatitis because we didn't even know what causes that. And in 89, Harvey Alter discovered the hepatitis C and Michael Houghton. And then when I was like an early fellow, we started to have not me, but companies started the interferon therapy, which was like daily injections of interferon. And interferon makes people sick like if you have a really bad cold. And we were giving this like every day. And the response rate was about 30%, maybe 25%. And we were celebrating if somebody kind of after 6 months would maybe get rid of the virus. And it really wasn't until like 2005-ish or so when the Hep C replication system in vitro was established by a guy called Charlie Rice at the Rockefeller. And that really opened up the field because now we could go back and evaluate how this virus replicates, what are the components, and that rapidly led to discovery of these various serine protease and RNA polymerase inhibitors that are key elements in the viral replication. And it was like so amazing that in the liver meeting in 2014, these presentations came one after another from various companies that now all of a sudden we have like 97, 98, 99% cure, viral elimination. And so this is what we have now. If you have hepatitis C or you have a patient with that, we can treat people who have cirrhosis. We can treat de novo or recurrent hep C pretty much in most people with total elimination. I think it's a fabulous story, and this is something that's really so exciting. And I think hopefully this is going to be a future of medicine, provided that we survive the next 4 years. And so I think, oops, and I don't know where's my mouse. Oh, this one. Yeah. And so I just want to say that another Nobel went very appropriately to this, to those who participated in this, Harvey Alter, Michael Hartman, and Charlie Rice, who, again, it's just really rewarding to know these people and have worked with them. I think it's, fantastic. So anyway, I think looking ahead, there's a lot, at least in the field of the liver. The WHO actually has a global elimination goal for hepatitis B, which is another chronic viral infection. And then, of course, there are multiple components, particularly related to reversal of fibrosis and liver regeneration that we have to figure out for the future. And I just thought it's appropriate to kind of give gratitude to all the Hungarian kind of, training and the university. I'm from Debrecen originally, but I have a close tie to the Semmelweis also, and I really had the privilege of, being there and had multiple postdocs, and I think it was just absolutely fabulous. And it's just having a great time. So the other acknowledgement goes to my colleagues and some of our colleagues around the US, and to the National Institute on Alcohol and Alcoholism and Aging Institute that gives us money for all of this, at least today. So maybe I just stop here. Thank you.
Sorry, thanks. Thanks again. And so other than alcohol, what if we can't cut that out of our life, maybe just lower the amount, what are the other factors that we should be aware of? Because you showed like donuts and pizza and things like this. Like, are there obvious factors that it would be also easy to avoid, or?
Well, the healthy lifestyle, whatever that is, I think is the one, right? I mean, I think it's the data is very clear on the kind of obesity, insulin dependence, insulin resistance, and interaction of all of this. So, it comes back to that. And some of the new drugs, the GLP drugs, actually are working very well. I think I have a slide actually here. That's just my family. But this is a new clinical trial that was just recently published in this is not alcohol, the metabolic dysfunction associated liver disease, that the semaglutide actually had very nice effects. And that was an interim analysis of this. So I think, I'm not sure. It almost sounds like too good to be true, because this is kind of another miracle drug, all these components. And in the long run, we have to see what is their effect. But so far, so good, because certainly they work very well on losing weight. They work on insulin resistance and the kind of metabolic components. But it looks like that it also has a beneficial effect on alcohol. So just do your exercise and drink moderately. It's okay. I think it's fine. No, don't think GLP. Don't get to that. What if you had to? Yes.
So the cancer risk that goes up with is that because of inflammation?
So that's a good point. Good point. It's probably way more multifactorial. Alcohol depends on which cell types you look at, could have direct effects on methylation and some of the sort of epigenetic factors that probably has a major role in there. And the inflammation in any cancer, we know that it's not a good thing, right? Because it just increases the kind of survival of cancer cells, and also promotes the or say impairs the natural recognition of the cancer cell that your immune system would eliminate otherwise, right? So we know that in
gut barrier becomes weak. You have more bacteria leaking into your blood that could trigger
absolutely
active immune response. Also, what happens to the T cells?
So
they have some
T cells.
active immune system.
So no, because what happens is that these antigen presenting cells actually are not working very well. So the T cell activation is really not happening. So in this alcohol associated, I showed you those neutrophils that in alcoholic liver disease, we have some early data that we haven't published yet that if you take those neutrophils and co-culture it with T cells while you are inducing T cell proliferation with like CD20, CD3 cross-linking, some other things, then actually the neutrophil, these alcohol-induced patients' neutrophils reduce the T cell proliferation. So they have some immunoinhibitory effects. And then this is something that actually we showed like a long time ago in trauma patients, that those who had alcohol and trauma were kind of not responding well. I think you were first. Sorry. Somebody left you.
Yeah, just a quick question. Have you discovered any positive correlation of moderated alcohol drinking, like red wine? There is a myth around red wine.
Yeah.
One glass.
So I took out that slide that this is what, because we started out actually some of these alcohol studies in humans only. And in vitro systems, so that was at the time when there was all this epidemiologic data in man, had a moderate, the U-shaped curve, right? That if people don't drink, then they die more, but if you drink a little bit, then it kind of really helps and so forth. Probably it could be some truth to it, because we had in vitro data, again, inflammatory cells, but actually if you give them one dose of alcohol, that has some tolerogenic effect to the LPS-induced response, which is a major inducer of inflammation, but that kind of goes away after a while. But then if we keep giving alcohol every day for about 5 days, then the whole thing switches and then it augments inflammation. So we worked out molecular mechanisms for that through the NF-kappa B and whatever I-kappa B pathway. So I think it's yet to be seen. People who work in cancer, they claim that even the small amount is not good. And I think particularly for breast cancer, there are some studies in particularly in women. So there was one here, and then I sorry.
My question was almost the same. So my question is just outside the liver. Is there whether worth of anything now?
Oh, the resveratrol. So that was the question about the red wine. Well, I mean, I don't know the exact amount, but you would probably have to drink like, multiple gallons of red wine a day to get the beneficial effect of the resveratrol. So I would say the answer is no. But if it makes you feel better, then go for it. Yeah, sorry.
Thank you so much. Such an interesting information, and I have nothing to do with.
Thank you.
So you're coming from a total amateur, but there are so many products on the market today that are sold to people with the promise that they will, if you take them before drinking alcohol, they are going to prevent hangovers.
Oh, really?
So I guess one of my questions is, do they actually work? And the other question is, do you think, so you talked a lot about, and so is it possible to sort of go the prevention route where we are giving these to people who are struggling with alcohol, consumption, and be able to sort of mitigate the negative effects of drinking alcohol?
So to be honest with you, I'm not familiar with those kind of remedies that are out there in the market. I mean, theoretically, I mean, I don't know. I still don't know that if there is something that's not gonna prevent alcohol being absorbed. But recently, actually, there is data that alcohol metabolism happens in the intestinal epithelial cells too, not just in the liver. So I don't know. I know it's interesting because in the old days when I got into this field, there were these people talking about the what is the relation of alcohol compared to the food intake. And I think in the European, old type of European culture, a lot of time people had these fatty meals and then they were drinking mostly during the meal and after. And, coming to the US, there is a lot of have a reception with no food and drink, which obviously results in a much higher and faster alcohol levels. So, based on what we know about metabolism, maybe that is kind of a factor. So there's this sort of, there was certainly a component in there. Yeah. Yes. Oh my goodness. I'm having a great time. So maybe we start here.
Yes. Thank you very much for the really interesting talk. You showed a number of inflammatory pathways that are involved in mediating the alcohol's effect, and several molecular targets associated with these inflammatory pathways. So in your opinion, which one is the most promising target, or is it possible that you can only, would that be possible to interfere with only one pathway and decide?
So that's kind of the $1 million question, right? So out of these various cytokines and pathways, so tumor necrosis factor alpha is one that's increased, and it is major inflammatory cytokine. So back quite a while ago, when the blocking antibody like Remicade came available, there was a small clinical trial that they did at the Mayo Clinic in patients with alcohol-associated liver disease, and essentially after about 8 patients, they stopped because patients started to die there was increased infection in them. So with this whole increased susceptibility to infection and kind of reduced host defense, there's this kind of really fine line that you want to reduce inflammation, but not to the point that they can't respond. So that's why we thought that interleukin-1 is going to be the good kind of target, because it is like a secondary fuel for inflammation. It doesn't cut everything down, but so far it didn't work out. So it's going to be more complex. Yeah. Okay, then maybe go there and then back here. Yes, in the white shirt. I'm sorry, I don't know your name.
Yes. Thank you for starting the discussion. So we have a leaky gut, and there must be some neoantigens and what are there, but there is a T cell function problem. So in some are chronic alcohol users better or first responders to checkpoint inhibitors?
Good point. That's the experiment that I told my postdoc to do as soon as possible. Because we do find, so in this, so in this study we find like a myeloid-derived suppressor selectivity, and we find increased PD-1 and some of the other immunology molecules. So one of the things we want to do is to actually check that. The interesting thing is that, at least in mice, we also find that if we give like 4 binges of alcohol and then let them be for a week and come back and challenge them with LPS, they still remember and have a different response than if they never had alcohol. So it's kind of trained immunity kind of thing. But certainly the PD-1 is something that we find. We haven't done the experiment yet. That's the point. One more. And I think you had it here, or listen this. I'm going to be around so you can listen.
It was fascinating to see the inflammatory pathways that you were showing. I'm working in neurodegenerative and neuromuscular diseases, and I see the same pathways. And they have tried, pharmaceutical companies tried lots of drugs to try to reduce inflammation in these patients. None of them worked. So I was wondering, this is probably a consequence, not a cause. Do you think it's still worthwhile trying to mitigate the consequence instead of trying to tackle the cause of the inflammation?
Well, so in the case of alcohol, it's simple: don't drink, and there is no inflammation, right? So, obviously, in other diseases where the cause is not known, it's more difficult. And then when you have multifactorial components, that is more difficult. So, well, I mean, based on some of the data that we have, I think I could make the argument that even if you reduce the inflammation, it doesn't necessarily take the whole thing away, but at least attenuates the disease. And, we see that with neuroinflammation. I mean, we have a project that is kind of asking the question that in Alzheimer's disease, is alcohol consumption making it worse or better? And the answer is not as simple as I thought it would be, as welcome to science, right? At least not in mice. So I'm going to be around if you have any questions. And thank you so much.